Electron micrograph pictures of some organelles, membranous and non-membranous. Membranous organelles. Rough endoplasmic reticulum appears as parallel cisternae with ribosomes attached to their outer surfaces. Mitochondria appear as spherical structures of variable sizes and shapes. Mitochondria and rough endoplasmic reticulum. Mitochondria appear spherical, bounded by two membranes. The inner one is folded to form cristae. The mitochondrial cavity is filled with matrix, rich in dense granules, matrix granules. Rough endoplasmic reticulum, on the other hand, appears as parallel cisternae or tubules in longitudinal sections and as circles in cross sections. An example of non-membranous organelles, centrioles. There are a pair of centrioles present close to the cell membrane between the nucleus and the free surface of the cell. They appear in longitudinal sections as two cylinders perpendicular to each other. Notice the presence of microvilli on the free surface of the cell. Transverse section, on the other hand, showing that the wall of each centriole is composed of 9 peripheral bundles. Each is formed of 3 fused microtubules, triplets. Electron micrograph of some membranous organelles. Rough endoplasmic reticulum appears as parallel cisternae with ribosomes attached to their outer surfaces. Mitochondria appear as spherical and oval structures of variable sizes and shapes. Mitochondria and rough endoplasmic reticulum. Mitochondria appear oval, bounded by two membranes. The inner membrane is folded to form cristae. The mitochondrial cavity is filled with matrix, rich in electron-dense granules, matrix granules. Rough endoplasmic reticulum, on the other hand, appears as parallel cisternae or tubules in longitudinal sections and as circles in transverse sections. Electron micrograph of rough endoplasmic reticulum. It appears as parallel cisternae with ribosomes attached to their outer surfaces in longitudinal section profiles and as circles studded with ribosomes in transverse section profiles. Electron micrograph of mitochondria. Each one is enclosed by two membranes. The outer membrane is smooth while the inner one is folded to form cristae. The cavity of mitochondria is filled with mitochondrial matrix, rich in electron-dense granules, matrix granules. Rough endoplasmic reticulum in the lower part of the photo appears as parallel cisternae or tubules with ribosomes attached to their outer surfaces in longitudinal sections and as circles studded with ribosomes in transverse sections. Electron micrograph of mitochondria. Mitochondria show a variety of shapes, oval, round, irregular, as well as sizes, large or small, but all are enclosed in two membranes. The outer membrane is smooth, while the inner one is folded to form cristae of different shapes and sizes. Electron micrograph pictures of some non-membranous organelles, centrioles, both longitudinal and transverse sections. There are a pair of centrioles in each dividing cell. appearing in longitudinal sections as two cylinders perpendicular to each other, close to the cell membrane. Notice the presence of microvilli on the free surface of the cells. In transverse section, each centriole is formed of 9 peripheral bundles, each composed of 3 fused microtubules. Electron micrograph photo of longitudinal section of centrioles. The cell membrane of two cells are seen with projecting microvilli from the free surface. Centrioles in the lower cell appear as two cylinders perpendicular to each other in the upper part of the cell close to the cell membrane. Electron micrograph photo of a cross-section of a centriole, showing the wall is formed of 9 peripheral bundles. Each bundle is formed of 3 fused microtubules, triplets. Thus, the wall is formed of 27 microtubules. Electron micrograph pictures of some inclusions. Fat droplets or lipid droplets appear. Electron-dense rounded droplets of variable sizes. Melanin pigments or melanosomes are black pigments present in melanocytes. Lipofuscin pigment deposits are heterogeneous in appearance and thought to represent undigestible residues of lysosomal activity, secondary lysosomes. Electron micrograph of fat droplets or lipid droplets appear. Black rounded droplets of variable sizes may be large or small. Electron micrograph of melanin pigments in melanosomes. Melanin is present in membrane-bound granules called melanosomes. It is formed and found in melanocytes. Melanosomes appear as small multiple black granules scattered in melanocytes. Electron micrograph of lipofuscin pigment deposits. They are electron-dense membranous vesicles, very heterogeneous in appearance, increase with the advance of age, and represent end products of lysosomal activity.